Hi, my name is Amitesh Pratap. I work at Informatics as a machine learning fellow. And today I'm going to be talking about an information theoretic approach to improving the training efficiency of clinical diagnostic classifiers. Machine learning classifiers can be used to diagnose acute infections using patients' gene expression data. Such classifiers are typically trained on sparse clinical data obtained from multiple different data sets. This leads to high level of uncertainty in predicting the infection status of the patient. On the top left panel here, I'm showing a typical machine learning binary classifier trained on sparse clinical data. And in the middle, you can see a blue region, which is the confidence interval of the model, also known as the uncertainty region. And in the middle of that is a dashed line, which is the decision curve. So on either side of the decision curve is your infection status, either infected or non-infected. Now, once this model has been trained and new data has been introduced at the test stage, you can see the data points landing on the uncertainty region lead to misspecification of the labels. And that is those data points are shown by the black boxes. However, the data points which are further away from this uncertain region are classified correctly. Now, to improve the accuracy and generalizability of machine learning classifiers, often as the new data comes in, these machine learning models are retrained by augmenting the training data set with the new data. So this is shown in the bottom left and the bottom right panel. Now, I'd like to point out that the utility of the new data point in terms of improving classifier is not the same for each data point, by which I mean the data points which are further away, the new data points which are further away from the uncertain region, the blue region in the previous case, lead to very modest decrease in prediction uncertainty as shown. On the other hand, the new data points which are closer to the uncertain region lead to very large reduction in the prediction uncertainty, and thereby they help decrease the uncertainty in the model and lead to higher accuracy of the machine learning classifier. Gaussian process classifiers can be used to quantify the uncertainty in the prediction of the infection status for the patients. On the top left panel here, I'm showing prediction labels which have been correctly identified, and the blue and the yellow curves are the probabilities for each of those data points. And as you can see, the points which are furthest away from the uncertainty region are confidently classified by the GPC. However, the points which are closer to the uncertainty region, although properly classified, are less certain, as you would expect. Similarly, on the right panel, those labels are misclassified and they largely lie on the uncertain region, as we'd expect. Now, typically, the hyperparameters of these Gaussian processes are maximized using marginal likelihood, but we can also trace back the uncertainty region using the standard deviation and the posterior distribution of the hyperparameters. Now, these posterior distribution of the hyperparameters can be calculated using various techniques, including variational inference. But either way, these Gaussian process classifiers fail to tell us how, by acquiring new data points in which positions, would help us decreasing this prediction uncertainty and improving the classifier accuracy. Information theoretic framework can be used to identify the most optimal new data points, which when added to the existing training data set will lead to the largest reduction in the prediction uncertainty. Specifically, the gene expression data points from patients which lead to maximizing of the mutual information between the infection status of those patients and the hyperparameters of the Gaussian process classifiers would lead to the highest reduction in the prediction uncertainty. Now, furthermore, mutual information also predicts these optimal data points close to the uncertainty region. And now, if the new data points from patients are acquired closest to these optimal data points, then this would automatically lead to reduction in the standard deviation of the posteriors as shown in the bottom two panels. Now, therefore, instead of maximizing marginal likelihood, which is traditionally done for Gaussian process classifiers, we can maximize the mutual information and use that to predict these optimal data points at the same time. And at the same time, we can also find the posteriors of the hyperparameters. Using our information theoretic approach, we can design the most optimal clinical trial given the limited resources. We propose using k-nearest neighbors metric to map the optimal data points to their nearest neighbors in the existing data set. The demographic feature of these nearest neighbors, such as age, sex, race, ethnicity, etc., can then be used to design the profiles for the participants of a future clinical trial. Now, furthermore, we also propose updating the hyperparameter priors with their respective posteriors after each clinical trial. So, in effect, we are proposing a scheme where, if possible, instead of investing all the resources into one big trial, we split that trial into multiple smaller trials, and after each of those trials, the priors get updated by the respective posteriors, thereby allowing the most informative dataset to be gathered through this process. And finally, I'd like to thank Michael Mayhew from Informatics, and also the computational biology and the machine learning group at Informatics. And also, thank you for listening.